Today, I'm very excited to have Hannes Stark joining us. Hannes is a PhD student at MIT, advised by Regina Barzile and Tommy Yakola, working on generative models for simulating and designing biomolecules. And he is known for BoltzGen, and he's going to tell us about BoltzGen. Great. Take it away.
Okay, yeah. I'm very happy and excited here to be presenting BoltzGen on behalf of our team. And what we're doing in this binder design model is that, for example, we have here in blue the target that we're calling it third telomerase, which our collaborators from the Dana-Farber Cancer Institute have identified as an important target for glioblastoma cancer cells. And what this protein does in glioblastoma cancer cells is that it repairs this DNA that you see here on the right of the blue protein. And then it helps the cells to survive, the cancer cells to survive their rapid division and growth. And now what we want to do is to design this binder here in yellow that will then displace the DNA and then prevent the repair of the DNA and thus then prevent the survival of the cancer cells. And now, if we wanted to do this, then maybe you already know some existing models for this binder design task, but they would fall short here in that first, they do not generalize to novel targets in the sense of the telomerase here, where we do not already have an existing protein that is bound to this target protein. And second, they do not support the constraints that our collaborators want to impose on the designs here. And now this is what we want to do with BoltzGen. First, on this axis, or the vertical axis, so to say, we want to have one model that can design us nanobodies, proteins, peptides, all different types of biomolecules, and then also target all different types of biomolecules, like a small molecule, DNA, a protein, maybe disordered regions in a protein. And then the second thing on the horizontal axis, what we want to achieve is to also generalize to novel targets in the sense of targets that do not have already existing bound structures in the training data, because this is indeed how most existing models have been evaluated, where we usually test them on targets that have already been in the training data with existing bound structures. And these two aims of generalization and um, applicability across domains, they go hand in hand, because we want to learn for generalization to be able to expand to novel targets. We want to learn transferable patterns like physical chemical interactions and 3D geometry. And these patterns are conserved across molecular types, molecular classes, such as, for example, the hydrogen bonds that make up DNA-based pairing. They are also the hydrogen bonds that stabilize a DNA helix. So training on all these different modalities isn't just nice for having a model that is widely applicable across all different modalities, but it also helps us and aids us further toward this task or this goal of learning generalizable patterns that help us in generalization. And now I want to take away, before talking more about the model, that to test the model, then we need to bring the designs into the real world and make them physically and test them in the wet lab. And for this, we now collaborate with more than 30 wet labs all over the world. And I'm happy that later I'll be able to share a subset of our results across 8 wet lab campaigns and 26 targets. But now back to the model. So the first piece, strong structure reasoning, what I want to talk about there is that um, I'll get into it a bit more detail with some additional slides, but just saying that if we want to be good at designing strong binders in a structure-based fashion, then we need to be strong at reasoning about structure. And second, we want to sort of get closer toward emulating physics, and then we make some arguments for how our training scheme or distribution helps us with that. And third, we want to allow the user to fully control the designs in their desired fashion. And first, towards strong structure reasoning, the point there that we're making is that if we want to do design, right, then this means discriminating between different options for what the binder could be. Here on the right, we're like saying, if we want to decide, will A bind to B, will A bind to C and D, and then choose between these options B, C, D, then this means understanding how A would interact with B, C, D, because we need to discriminate between them, right? And this understanding how things interact, that is the folding task. And right, in other words, if we want to be strong at structure-based binder design, then we need to be strong at reasoning about structure. And the point is toward emulating physics I already made, that the patterns that we want to learn, because they generalize to novel targets, they are also then the patterns that are general across molecular modalities, like physical interactions. And then just one example for controllability, what I mean with that is that sometimes our collaborators want us to design only these orange regions here in a protein, and sometimes they want us to redesign everything. And this is, so to say, the spectrum of possibilities for this partial design scenario, but this spectrum of possibilities also exists for binding sites or covalent bonds that the user might want to specify. And then the point number three, this or first back to point number one. Now, how do we achieve this strong structure reasoning? And before I get into that, I want to take away that this diffusion model that I'm talking about now, it is just one piece of a larger pipeline, and maybe the most important step of this larger pipeline, but the other steps are also crucial to then end up with good designs. And now what we're looking at here to understand how this first step of the larger pipeline works, this diffusion model, is one denoising trajectory where we're generating in yellow the binder to the target in blue. And we're starting from complete noise for both of them. And then for this target, we're reasoning about how it changes its conformation, and we're folding it while understanding or how it will change its conformation under binding the binder that is simultaneously being designed to bind to the target, right? And this then means that for the target, we're just predicting the structure. We're just generating a fixed set of 3D coordinates for every single atom, because we know how many atoms there will be in the target, right? We know what our target is. But for the binder, we're simultaneously generating 3D coordinates and a discrete sequence that describes this binder, right? Because this protein binder consists, or its identity is described by a 3D structure and the sequence. And now there's a little issue because, right, if we don't know what the amino acid types are yet, because we don't know what the discrete sequence is, then we also don't know how many 3D coordinates there are. So the dimensionality of the thing that we're generating, it changes over the course of generation with how our belief of what the discrete sequence should be changes. So we're having this dimensionality changing thing where we don't know how many 3D coordinates we even want to generate in the end. And how models usually would deal with this is to just ignore these side chains, they're called, of residues, and only model the atoms that are shared between all different possible amino acid types. And this then means that the model reasons about some abstract amino acid types and an implicit meaning for what they could be, instead of reasoning about atom interactions. And also, the designed residues, they're treated very separately and differently than the residues in the target. And now we want to get away from this and have an explicit treatment where we sort of solve this or bring this two together by embedding the discrete residue type choice into the continuous coordinate generation. So we represent both the discrete denoising process and the continuous denoising process in one space. And this is this 3D space, whereby we then say that just every residue for us now has always 14 atoms, where some of these atoms don't correspond to real atoms. And then the geometry that we generate, it determines what the residue type is. And this way, then the model will not be reasoning about some abstract logics or implicit representations of what these residue types would be, and what their meaning is, but rather, for example, if we're generating a tyrosine, like this tyrosine in the middle, then we would be placing an aromatic ring there. And then also, if we have the belief or the model carries out the belief during denoising that there should be this aromatic ring here, right? Then we can still modify this because maybe it should be an aromatic ring without this additional oxygen, and we want to have the phenylalanine on the left. And yeah, we're reasoning about these atoms explicitly. And this then also means that we're only generating 3D coordinates for the binder and 3D coordinates for the target. And this is very nice because then we can just have one unified model for both structure prediction and for binder design. And the features that we learn for structure predictions are directly useful and transferable for the binder design task as well. And this is all that I wanted to say about this concept of unifying design and structure prediction, because this now means that we also can train on both tasks simultaneously, right? And now to the second point of training across how we exercise the model in many different contexts to try to or to encourage or towards the only viable solution being reasoning about transferable patterns. And I'm to explain how this works, I'll just step through an example of how a training step would look like. So we would have this complex here on the left from PDB, for example, and then we take a crop, and crop is random and of a random size. And it is centered or random also in the sense that it is centered around this residue that is picked randomly. And then we have this substructure that we see on the right here. And then we train with multiple possible tasks on that substructure, where first, for example, in blue, we're just predicting its structure. The model gets the sequences input. There's no design going on at all. And the model just predicts the structure of the protein, the 3D coordinates. Or then sometimes we train with a design task where we say for the blue area, you get the sequence as input again, and please predict the structure. And for the yellow area, you do not get any sequences input, and please produce this new 3D structure where we have this sort of augmented representation where each residue has 14 atoms that encode their geometry and their residue types. And then sometimes we feed the structure as inputs. And sometimes we feed with a certain probability a binding site specification as input that the model then will hopefully adhere to at inference time if we want to specify where the binder should bind to. And these distributions, they also change, or these probabilities with which tasks we train, they also change over the course of the training. Like, for example, at the beginning, we only train with structure prediction. Okay, but that's it, and I will just
Can you go
briefly.
back to the previous page? Like, what about the latter two tasks, structure input and binding side input? Like, are those probabilities just illustrative or like somehow, like, I guess I'm just a little confused how you ordered between these four tasks.
Yeah, so these are 4 tasks of 20 or 30,
Yes.
right? So I'm just laying out a few here. And now these probabilities that you're seeing here, it's not like they should add up to one. It's when we're doing structure prediction, when we only train with structure prediction, then the latter two tasks here are not happening at all. Only if we're doing design, then we sometimes in the design scenario then go into the next part of the tree, so to say, and then with some probability feed the structures input, with some probability feed the binding site as input. Yeah, maybe I should make this a bit more clearer. The sort of yeah, it's a bit confusing that this stuff doesn't add up to one here. Okay, but then what are the different control options that we have on the model? So we already encountered a scenario where we can say, right, we want this to be a cysteine, this other the orange thing to be a cysteine, and then only the other residues are up to the model to design. And then we can also control it by saying we want the thing to be a helix or a sheet and determining its secondary structure. You can feed in covalent bonds to control the structure. You can specify what you want to be the binding site, what do you want to be not a binding site. You can say sort of control the structure in some quite fine-grained manner, where you can say that a certain region you don't want the structure to be predetermined, in a certain region you want it to be predetermined, but only with respect to a certain other set of residues. And then, for example, here, the region that has visibility 2 and the region that has visibility 1, they're fixed within themselves, but their relative position is up to the model to decide. And all these control options, so to say, they're fed or combined together into sort of a design specification language with which you can decide what the object is that you want to generate. And now I will, or this, you have this design specification, right? And then you feed it as input to our generative model, this diffusion model, and then it produces something like 60,000 designs. And then you optionally inverse fold them. And if some of you know inverse folding models, this is just like our version of soluble MPNN. But if you don't, you can just see it as we refine or we redo the sequence of the design binder while keeping the structure. And then we refold it where we extract the sequence from our design structure. And then we refold it with some structure prediction model, Boltz-2. And then we check, does the refolded structure match the structure that we designed? And we hope that to be the case and use this to later filter. And then we compute sometimes an affinity prediction with a computational model, which I'm skipping here now. And then here in this analyze step, we are computing these counts of physical interactions like salt bridges, hydrogen bonds, and we're measuring how much interaction surface area there is, for example, between the design protein and the target protein. And then all these metrics are fed into a final ranking algorithm that combines them into a single ranking. And then from the single ranking, we run a quality diversity optimization algorithm to balance sort of this quality and having high ranks with also having high diversity in the binder that we designed. And then we obtain the final set of designs of like 15, for example, that we test in the wet lab. And we'll get to these wet lab results now, but before that, I will briefly show you some results that are purely computational, and we're looking at some fun animations. But these computational results are now not what you maybe have seen, this designability numbers that people often look at in these types of binder design papers or unconditional protein structure design papers. We ignore this metric completely, and we rather optimize our model for, well, being able to do design, but then we optimize it for being good at predicting structure, for being good at structure prediction, because we think that this is what you want to optimize for. You want your model just to be good at reasoning about structure rather than these proxy metrics of designability. And then the metric that we're looking at here shows us that our or in blue, BoltzGen's structure prediction performance, right? Our design model's structure prediction performance is equivalent to that of a pure structure prediction model, Boltz-2. And this holds also across modalities. But now to the real wet lab results, where I mentioned these now more than 30 collaborators, and that we have the results from 8 wet lab campaigns and across 26 targets. But I will first, or I won't show all of them, just a few. And starting off with our campaign where we try to find binders against novel targets in the sense of them not being targets that are already in the training data with existing bound structures, right? Which is, as I mentioned, most models, most binder design models are evaluated at the moment, right? That they
Sorry to interrupt. I'm not too familiar with the field, but how does your generation connect to the wet lab exactly?
So how is my generation collected to the wet lab?
Connected to the wet
Ah,
lab? Like how do wetlab exactly use your generation, and then
Yep,
how do they have machines to produce these proteins directly, or like how
We
does
will,
the
yeah, we will see this on the next slide. So
okay, sounds
um,
good.
we now, right, what we want is that we have our target protein here. So in blue, we have now the targets that we chose. And usually what people do is that they take as target a protein that is already in the training data with an existing bound structure. And now we make sure that this is not the case by taking some quite dissimilar proteins to anything else in PDB that already has a bound structure. And then we test on them. So we assume that this should be quite a lot harder evaluation. Then what we evaluate is that we now generate these 15 designs with our model, and then we send them to this wet lab, which tests do the proteins that we produce even express, do they exist? And then they test, do they bind to each other? And this binding is measured in this KD number that we want to be as low as possible. So here it's like 6.1 to the times 10 to the power minus 9, right? So 6.1 nanomolar is this KD number. And what it describes is the ratio of the two things detaching from each other divided by the ratio of the two things attaching to each other, right? And we wanted to detach very little or very few detachments per second happening, and we want to have a lot of attachments per second happening. So this off rate divided by on rate, and that is this KD number. And this, of course, we then want to be as low as possible because we're dividing the off rate by the on rate, and we want a high on rate and low off rate. And that is what is measured in these wet lab tests here. And then we find that against 6 of the 9 targets, we find nanomolar binders, where if as long as something is a nanomolar, it's kind of considered good, and low nanomolar is considered quite good. And if you have sub-nanomolar, it's considered very good. And yeah, in comparison, like if we look at RF diffusion antibodies, for example, they have targets that are in the training data with existing bound structures, and then they, for example, test 9,000 designs instead of testing 15, and then obtain, in 2 cases, micromolar binders, so 1,000 times weaker than what we see here. And in one case, they also obtain nanomolar binders. Okay, and we also then test this design that we make, the protein that we designed, this nanobody, does it actually bind to the target or does it just bind to anything? And we test this with taking another target, HSA, human serum albumin, which is just very interactive and tends to stick to many proteins. And then we see that our protein or our nanobody actually only binds to our target and not to this potential off target. And then we also do this for not designing nanobodies, but just vanilla proteins and see very similar results. And then we have some interesting cases like this one here, where we have this NUP98 protein, and this forms a fusion, or it, I won't go into that, but the NUP98 protein, it can have a mutation where then it causes these what's called condensates in a cell. What you can see are these purple dots in the top right image. And then these condensates, they recruit some proteins, like some growth factors maybe, into the nucleus of the cell and then cause the cancer, cause leukemia. And thus, we want to design this peptide here that binds at this region and then dissolves the condensate by preventing the NUP98 from binding to each other. And we tested 9 binders for whether this happens. And for one of them, this was successful. And this model is now going into some actual cancer cell line models where they're testing, will this peptide then help against this cancer? Okay, and then we also have other evaluations like designing proteins that bind to peptides, where we're quite successful, or we design proteins to bind to small molecules, where we also have like very high success rates that haven't been previously seen. And we're, for example, able to get nanomolar binders right away while just testing 3 designs against a certain target. And that's where I want to conclude with saying, right, we set out on this task to improve generalization or enable these models to also generalize to new targets and to generalize across modalities like proteins, peptides, nanobodies. And then we had this task or this solution where we unify folding and binder design in one training procedure and one model, and then also across all different possible modalities of biomolecules. And then we have this design specification language with which we can control what we're generating, because we, for example, want to be able to specify binding sites, or that we generate a nanobody instead of a vanilla protein. And then we have some experimental validation with many wet labs all over the world, and across 26 targets. And with that, I want to say that we maybe now have a model that is fine at designing binders, and also at that there are still many limitations, right, for designing binders. But if you want a therapeutic, then we also need more than to just design a binder, because we need to check or also build into our modeling that the thing that we're designing doesn't have any off-target interactions, and it's not detected by the body as something bad and being attacked by the body, and needs to be delivered to the right location, and so on and so forth. And of course, we're working on that next to many other problems, like, for example, designing also aptamers, like RNA, to bind to proteins. And if you want to get involved in any of that, I would be excited to talk about it. For example, in the selectivity-aware generation space, there are very exciting questions about constraints or compositional sampling of diffusion models, where you now want to combine your diffusion model that samples the distribution of binding to target A, your diffusion model that samples the distribution of binding to target B, and then how can you combine them to sample the distribution of binding to A and not binding to B? What is even the right formulation of the distribution that we want to sample there? And yeah, with that, I want to conclude, and all the links to resources and various things are here. And if we have any questions, I will take them.
Cool. Thank you so much. I'm pretty new to the field. I'm curious, like, you did to learn a little more about the training details of the model. Like, I think you explained the model, but what is the training data? And I guess, like, you mentioned a lot of future directions, but I guess, like, maybe just on the model itself, like, what do you think are the most important future directions, or do you think the model is good enough already and it's more about, like, testing other kind of things like affinity and so on?
Yeah. So this training procedure, what is the training data? Right? It's just these 3D structures of proteins. There we're looking at it. We have this protein on the left here, where we have this. This is a 3D structure, right? And it has a every 3D or every segment or little segments of the 3D structure also have the sequence identity associated with it, like what amino acid type it has. And then we're trying to just predict the structure of this 3D object given the sequence, or given the molecular identity that's described by its sequence, or we're trying to predict the structure of one piece, but then simultaneously design a structure and a sequence that would bind to that piece. And does that sort of explain the?
That still feels to me like the model part. Like, I guess I'm curious, like, just like what do we already have? I assume with AlphaFold we do have a pretty good database of structures.
Okay,
But,
I
like,
see. So
this kind of binding data. So, like, yeah, I'm trying to understand more on the data side of, like, what is
the main
used.
training data set here is PDB, and this is a agglomeration of, or This is a collection of all protein structures that are known to humanity and that are experimentally resolved. And these are like collected over 30 years or so, and we have around 200,000 of these structures. And then the other source of training data, where I do not know if this actually helps or not, is a set of monomer structures, so only single protein chains and not any binders in there, that are predicted by AlphaFold, yes. And then only the ones where AlphaFold has very high confidence in its structure prediction, those are additionally used for training. But there we're not sure if this additional training data even actually helps. And it's like we don't have much evidence that it helps, but just from our prior, we still that it should help, we include it in the training data.
And for PDB, it already has like part of the data that's binded protein. Like maybe I just don't understand the protein enough. Like,
Yeah.
are all the protein structures in PDB useful for the second part of your like model training, where you need to design as well in addition to do structure prediction?
Yeah, we can just flip through here a little bit, right?
I don't think you are sharing
Oh,
screen.
oops, oh, shit. That was a problem. Then, yeah, here, right, we are looking at this database, PDB, and there you can see like a standard structure here. If we just look at it, it has a protein chain bound to another protein chain, and there we would learn about these binary interactions. But we also learn about interactions from just a single structure, right? Because there are also interactions happening inside of a structure that contains only a single protein. Yeah. Like this here, for example, it's a single structure, but we would also see how this helix here is attached to the sheet and they stick together and learn all these aspects.
I'm also very curious about. Oh, sorry. Go ahead.
No, I was just wondering what other questions are.
Oh, yeah. I'm just very curious about how these things apply to real applications. Like, for example, when you generate those proteins, do you have almost like a target property in mind so that you want to achieve, or do you
Yes, we
say
do.
in the binding stage, do you generate and then test how it binds with like a large collection of proteins so that it kind of reflects what it will happen in real life? Is that part of pipeline?
What will happen? Like, right, we are trying to design something that binds, and that is all. We're only trying to design binders, and binding means that the stuff sticks together. And now, then, why do we want this? Because, for example, a case like the example in the beginning, where usually the DNA would be repaired. But now, if we have this yellow thing here that we designed that sticks there, that binds stronger than the DNA does, then the DNA can no longer be repaired. And then the cancer cells no longer survive because their DNA is no longer being repaired. And
Interesting.
then we have what we want, and this is maybe a drug and helps us against glioblastoma cancer cells. And then, of course, we don't know if this is a binder. And to know if it is a binder, we need to test it in the wet lab.
I see. Very interesting. Thank you.
Oh, sorry, I'm going back to the model a little bit. Feel free to answer questions. I guess just like I'm pretty new to this, so I'm hoping to learn more about like how BoltzGen works. And you mentioned like how you encode them in the 3D space, but in the model, do you end up having components that's more like graph neural networks, or do you just have like long sequence of many 3D coordinates concatenated together?
So it is mostly like a transformer, what it looks like. It's operating on these 3D coordinates. So a transformer takes
So
in
you
the
didn't
3D
find like
coordinates.
explicitly modeling the structure is helpful. Like you just putting it as a long sequence and send that to transformers is already good enough.
Yes, that is what the architectures resulted in the end. That it is the identical architecture to AlphaFold. It is just the architecture of a structure prediction model. And there's no equivariance or so happening. No 3D, no SE3 invariance or equivariance.
Yeah, another high-level question I had is like in our lab we have this hypothesis generation project where we, generate some hypothesis about observed phenomena, which I think is a little similar to what you are doing. Like the way you generate those proteins can be also framed as, the hypothesis is this protein will fold. So I don't know, have you and in our case, the hypotheses are like less structured, or like it has less of that, domain expertise kind of aspect to it right now. The way we do things is that more like we just use a language model and then we pass in some observations and then let it sort of like summarize patterns as hypotheses. So, I'm just wondering, like, if we want to extend that to say what you guys are doing, do you think there's a way to almost like combine the two approaches where your system produces exact protein structures and then our system may do almost like this educational purpose where we summarize human interpretable patterns that we can almost teach scientists and guide
But
their
then for that, I think we would need to sort of first teach the LLM to understand protein structure, right?
yeah.
And somehow need to connect protein structure to human language and sort of train the model to, in human language, describe protein structure. For that, I don't know how you would construct a training corpus. I mean, there are some straight up ideas, but how to really get it working, I'm not sure if the straight up ideas already work. Maybe I'm not sure.
Yeah, I guess there are some efforts that people are trying to do, like training uh, like a almost like a chemistry language model or like some other different language model that takes domain
Yeah,
language, basically.
I know, I know.
Yeah, but I'm not too sure like there's any really good ones yet. But yeah, thank you.
Maybe so. There's this resource that just very recently came out. Lab Bench. Maybe if you know that. Maybe this is something somewhat interesting to look into in that space if you want to connect language models with
Yeah,
um, biology.
I see. It's from Future House. Yeah, they do a lot of biology, uh, AI for biology stuff. Yeah.
Mm-hmm.
Yeah, very interesting. Thank you.
Yeah, I guess if there are no other questions, let's all thank Hannes again. Thank you very much for taking the time, and this was very cool work, and I'm excited to see what comes out from the wetlab.
Amazing. Thanks
cool.
for the nice questions, and yeah, I hope the maybe hypothesis generation type of connections here, well, there might come something come off of it.
Yeah,
See you around.
sounds exciting. Thank you.
Bye.
Okay. Stop. Cool.